Vidhi madam, good afternoon.
Prishti sir, you can start.
Sir, thank you, sir. Sangeeta, am I audible?
Yes, sir.
So let's start.
Yes, sir.
Already
are honored to have with us a visionary leader whose commitment to academic excellence and innovation continues to elevate C.V. Raman Global University in global prominence. It is with great respect and admiration that I invite our revered chief guest, Professor Dr. Bansidhar Majhi Sir, Honorable Vice Chancellor C.V. Raman Global University, Odisha, to deliver the inaugural address and share his insights on the evolving role of digital technologies in pharmaceutical research. Sir, we are privileged to have you with us. The virtual podium is yours, sir.
Good afternoon. Good afternoon to all of you. Our distinguished guests, Dr. Umar Ranjan Lal, Major Mohali, Dean Pharmacy, Professor Prusty, other faculty colleagues, and my dear participant. In fact, I'm able to see there are almost close to 76 together here, including me also. Because participants are there around. Is a event that has been initiated by the Department of Pharmacy, C.V. Raman Global University, a webinar on digital frontiers in pharmaceutical research and innovation. Is a apt topic which is need of the hour. Dear participants, as all of you know that among the several SDGs, healthcare is one of the very important SDG out of this. And when we talk about healthcare, if the healthcare takes a front seat, and to achieve the wonderful healthcare or the better healthcare or the effective healthcare, pharmaceutical technologies and research, they also take the front seat to make it successful. So pharmacy plays very, very important about the effective healthcare. And when there are changes in the technology, there are changes in the environment, and we are in fact going to a more and more digital world. And as earlier all of you know that things were not happening that digitally, but almost all healthcare systems has become digitized, and we are able to get the digital data in our domain. And when we want to analyze, you want to use the technology. And as you know that when earlier IT people were talking about that they are novice or they are illiterate, but when those days are gone, when we talk nowadays that the people have to know AI, artificial intelligence. And when we talk artificial intelligence, in fact, it is a very, very broader technology playing a very, very vital role in all aspects of life, starting from manufacturing to the healthcare to the or research in other domains. So to the agriculture. So this AI is really revolutionizing so many things, and this pharmaceutical industry is also option to this. And this 6 days workshop, I'm sure there are people who are working in the domain. I'm basically a computer science professor, but work in AI. In fact, in biomedical image processing, I have worked a little bit. So I understand industries, these digital technologies, how important they are. And when you all participants must have some domain knowledge in pharmaceutical industry or might be undergoing research, some of you, or might be planning for further research. So it is essential to know the role of artificial and other digital technologies in pharmaceutical research and innovation. So I'm sure this is a really apt topic chosen by the department or the pharmacy faculty, so that it create a larger platform for several and the speakers which who are working. and it will give a wonderful experience both for the speakers as well as for the participants, so that these lot of quality deliberations can take place, and from both the sides, is both from the learner or the speaker side as well as from the participant side, there will be a great academically fruitful deliberations. And but I request that even if you are in person or the offline mode is much more effective, but online has become our new normal. So all of you should take advantage of this without being, even if you are online, please understand or please be attentive, uh, work in collaboration with the speakers so that it becomes an interactive session. This program ultimately becomes successful. With this note, once again, I wish all the participants an academically fruitful journey during the next 6 days. And this group or this particular or like team which has been created through this online platform must continue to interact even after collaboration. Thank you. Thank you very much.
Thank you so much, sir. Now it's time to hear from a distinguished personality whose contributions to pharmaceutical science and education continue to inspire excellence and innovation. We are deeply honored to have Dr. Umaranjan Lal with us today as the guest of honor for this national webinar on digital frontiers in pharmaceutical research and innovation. May I now respectively invite Dr. Umaranjan Lal to share his thoughts and insights with us. Sir, we are privileged to have with you. The stage is yours, sir. Sir?
Very good afternoon to one and all. I thank the. Should I start with the presentation? I thank the management for inviting me. I think I'm audible to all, Dr. Prusty. And
Audible, but sir, your video is not coming. I think I request all the wonderful. That's great.
So I thank the management for inviting me for this day one, and this. I will directly move to that topic. Just I will share my this. I think here it is coming, the host is not allowing multiple presenters sharing for this meeting. So kindly from your end, something has to be done.
You need to give permission to sir as a host. Yes.
Is it now okay, Dr. Uma?
It's coming, but
Sir, you have to click on that particular presentation because your desktop screen is visible.
okay.
Yeah. Now,
Now
make it full screen. Yeah.
I think
it's okay.
So very good afternoon to one and all, and I thank the management for inviting me. to give a lecture on the topic bioinformatics, pharmacoinformatics in Ayurveda. So I will just start with the introduction of bioinformatics, what exactly bioinformatics is. It is an emerging interdisciplinary term that refers to the application of computational, data science, and bioinformatics tools to Ayurveda. Okay, and it combines Ayurveda with informatics to explore and validate the Ayurvedic knowledge in a scientific and technology-driven framework. What exactly we are benefiting from application of pharmacoinformatics in Ayurveda is that we have a traditional wisdom where our traditional system of medicine is rooted very deeply, and we are using that for since many thousand years, and we combine the modern computational tools for the development of evidence-based phytopharmaceutical drugs. So let us try to understand what exactly informatics does. Informatics is a computational tool, and it analyzes the data, gives computational modeling, it's artificial intelligence, and system biology. In context of Ayurveda, it bridges the gap between traditional knowledge and modern science by digitalizing classical formulations, identifying active constituents, and predicting their therapeutic potential using the computational tools. So what are the key areas in bioinformatics that I have tried to jot it down? Digital Ayurvedic knowledge systems. So you must be aware about this Ayurvedic databases that are available. We have many texts that are now online. You can have Ayurvedic knowledge from Ayurvedic systems from that. Then phytochemical and pharmacological mapping. Using chemoinformatics and molecular modeling, Ayurvedic herbs, bioactive compounds can be predicted. And those compounds can be mapped using modern drug targets and pathways. Reverse pharmacology and systems biology. Since all formulations are multi-component, multi-targeted interactions of Ayurvedic formulations are done in bioinformatics. Another is we have network pharmacology to validate the traditional formulations. Then we have artificial intelligence machine learning in Ayurveda that predicts herb disease associations, personalized medicine based on Prakriti. Prakriti is the nature of an individual. It differs, it always differs. All persons are not same. And that's why pharmacovigilance came later, and we were aware with the Prakriti of all individuals, and the drugs for all persons were different. Okay, then diagnostic tools integrating Nadi Pariksha and biosensors, the standardization and their integration. Then again, bridging Ayurvedic classifications with modern biomedical descriptions, developing interoperable data standards for global research, and finally, drug discovery and their development. So this is just an example to show you how this in silico analysis can lead to development of phytopharmaceuticals. Just I have taken an example. There's a proposal like we can use classical formulations for the development of phytopharmaceuticals. I have given one example of Jiraka de Arista. Jiraka de Arista is a classical formulation used for Sutika Roga. Sutika Roga is a disease which is a postpartum period. After the childbirth, the female they have a lot of disorders. For that purpose, Jiraka de Arista is used. And is it used traditionally? We analyze the constituents present in Jiraka de Arista, like sources of the what are the marker constituents, how the formulation is prepared, and what is the fate of marker constituents during the preparations. We get down to those things after SPHA analysis. Then we perform in silico analysis and validate that with the modern techniques like cell line studies system. Here we will use MCF cell line and HERA cell line for the development of phytopharmaceutical. We find this, if the things are done in this way, it will be novel, it will be significant, and it will be non-trivial, as it is based on traditional system of medicine. For example, let us try to see again further. It is a multi-constituent formulation. It was prepared in-house, then HPLC analysis of that was done, and many constituents like luteolin, 7 glucosides, apigenin, 7, these are flavonoids, and they were found out in this formulation. So let us try to see how this is important. So as I told you, flavonoids and their glycosides were the major constituents. So you see, we have been using these formulations since ages. And if you compare those reports, recent reports on women health and flavonoids, you will find that the flavonoid apigenin, it modulates activity of uterus. Apigenin is a promising agent for enhancing female reproductive function and treating associated disorders. And further, another flavonoid, luteolin, which was another major constituent in the formulation, it causes vasorelaxation in uterine arteries from the normal pregnant girl. Means the things which we have been using for ages, now it is being validated by modern study. Okay, so the importance of AI is very important here. Based on the Ayurvedic formulations, using AI, we can develop good phytopharmaceutical agent. Okay, the problem with Ayurveda is that the formulations are not well standardized. So how the information from Ayurveda can be transferred to the AI so that better validation can be done. For this purpose, intervention of pharmacoinformatics is required in Ayurveda. So let us try to see what are the things that we are going to cover in this. So we will be seeing bioinformatics and molecular modeling about the small molecules and macromolecules, 1D, 2D, and 3D representation of small molecules, how to draw 2D and 3D structure of ligand, what is force field and energy minimization, how to prepare ligands, how to get information about macromolecules, how to select right PDB structure, docking tools, preparation of macromolecule for docking, grid generation for form molecular. These are the simple things that I will be today introducing to you all. So what exactly is bioinformatics? As I have told, I have just spoken. Bioinformatics is a specialized field that integrates principles of Ayurveda informatics for modern computational, using modern computational tools and techniques for advancing the traditional practice of Ayurvedic medicine. It involves application of data science, computational modeling, bioinformatics, and artificial intelligence to knowledge base of Ayurveda, the ancient system of medicine that originated in. Molecular docking is one of the tools of computational techniques utilized in modern biology, chemistry, and pharmaceutical sciences in Ayurveda. What molecular docking does, it is used to screen natural compounds in Ayurvedic herbs for their potential drug candidates, to analyze various Ayurvedic formulations and their composition, to get their mode of action, and to find out a specific druggable target for each formulation component. So they are the bioinformatics and applications in Ayurvedic formulations. So what are small molecules? So small molecules, scientists synthesizing a compound in the lab or phytochemical. Okay, heat. The compound is tested and shows activity against the disease, that is heat. What is lead? A compound is modified to improve its binding affinity, stability, and selectivity. What is a drug candidate? A drug candidate is the lead molecule that undergoes preclinical testing, showing promising results in animal models and favorable safety profile. What is a drug? After clinical trials and regulatory approval, the compound becomes a licensed drug available for patient treatment. These are the small things. So how the small molecules are represented? 1D representation, like haldi. From haldi we have curcumin. Molecular formula is C21H20O6. Okay. UPAC name, the same, this E and Z, that defines the stereochemistry. This means 2, and the diene means 2 double bonds are there, 3, 5 dione. and SMILES notation. I don't know how many of you have used this SMILE notation for drawing the structures in 1D. If you copy this SMILE notation and paste on ChemDraw with the special pasting, you need not to draw the structures. It will be automatically drawn. This is 1D representation. Coming to second 2D representation, it is a structural diagram where atoms are represented with symbols like circles or letters, and bond between them are shown as lines. You can see the structure of curcumin. It is enol form. One is keto form. So this is 2D representation. When it comes to 3D, you have the structure. You can have a 3D look of the structures with all atoms like designated as colorful, like black ones, carbon bonded is there. Red one is oxygen, and white one is protons. Okay, so this is 1D, 2D, and 3D representations. Now, how to draw the chemical structures using the sketcher tools? The left one is ChemDraw, where many toolbars are there. We have a status bar, BioDraw toolbar, menu bar, SciFinder, where you can search the structure using SciFinder, whether your compound is new or not. Text formatting tools are there. When you use this ChemDraw, you will be fully aware what I'm talking about. Then you have 2D structure. 2D structure you can draw using Researchand. Then coming to macromolecules. What are macromolecules? Macromolecules are large complex molecules that play a central role that is on structure and function of cells and organisms. A macromolecule may be a protein, nucleic acids, carbohydrates, and lipids. So few macromolecules are there: protein, enzymes, receptors, genes, and antibodies. All enzymes are proteins, but not all proteins are enzymes. This you have been hearing. Similarly, all receptors are proteins, but not all proteins are receptors. These are type of macromolecules. Now, what are the databases? How you can search the databases? You must have used PubMed, okay, where you can see the database for protein primary information. Then you have database for 3D structures of primary protein. When you will search on the net, these things will come, and using that you can search these things. What is a force field? Force field, in this presentation, I am trying to make you aware for the terms which are used in pharmacoinformatics. I am not going into much detail, okay? What is force field? Force field refers to the functional form and parameter sets used to describe the potential energy of system of particles, okay? A force field is a mathematical model used to calculate the potential energy of a system of atoms. It helps to predict how molecules behave based on their atomic structure and the interaction between the atoms. This is very simple, but these are the definitions of force field. So a force field in a computational chemistry typically includes several key types of interaction between the atoms, that is bonded and non-bonded. Clear? So how this energy minimization? Energy minimization is the process of adjusting the position of atoms in the molecules to find their stable arrangement. The one where molecule has the lowest energy. It is like finding the most comfortable way to arrange all the ball and stick model where all the atoms are at their ideal distances and angles, and no necessary tension or stress exists between them. So it is by manually we can using this ball and stick model, we can do it, but it is done with the help of computational method, and that is what energy minimization is. And this is done by using force field. Earlier I have explained what is force field, to calculate the energy at each possible configuration of atoms, and then gradually adjusting the position to reduce the energy. And what are those energies? You have M stands for molecular mechanics, BS stands for bond stretching energy, AB stands for angle bonding energy, TA stands for torsional energy, and van der Waals energy. Van der Waals energy, and again we have hydrogen bond other energies. Miscellaneous energy means hydrogen bond, this torsion energy, and other things. So these are the things used for energy minimization. This is pyridine. You can see how the molecule is arranged for this energy minimized. So you have another calculator which will show you another graph which will show you that at this particular point, at this angle, this particular molecule is having lowest energy. Further, you have something called graphical user interface or use interface of Researchand software. So this is the interface where you put your molecule and that minimizes the energy. You can have interaction with the protein. This is how the things are feeded in Researchand software. Researchand software. Let us try to see what exactly is docking. Docking is a computational technique that is used in molecular biology and drug discovery chemistry to predict how two molecules, typically a ligand as a drug or a small molecule that is a receptor, interact with each other. Docking. It will be clear. The interaction is usually studied to understand the binding affinity, orientation, specific interactions between the molecules, which is critical for drug discovery, enzyme substrate studies, and other molecular research applications. And these are the interactions I have talked just now. The ability of a protein and a nucleic acid to interact with the small molecules to supramolecular complex plays a major role in dynamics of the protein, which may enhance or inhibit its biological function. What are the modes? Respective, prospective. In respective modes, how does the molecule is binding? What is the mode of action? What might be the reaction mechanism? That is all is given. What is prospective mode? What compounds might be good leads? And what compounds should you make? That all information is given when you do this informatics studies of the ligand binding molecular docking studies. So this is just a pictorial representation. Like you have key, you have substrate, and that goes to active site, and that is how the things are done. It is frequently used to predict the binding orientation of the how docking. Why it is used? It is used to predict the binding orientation of the small molecules of the drug candidates to their protein target in order to predict affinity and the activity of small molecules. So how many types of docking are there? We have rigid docking, rigid ligand docking. In rigid docking, we have both ligands and receptors are rigid. Rigid ligand docking, receptor is rigid, ligand is flexible. Flexible, both ligand and receptors are flexible. Blind docking, no prior knowledge of binding site here. Induced fit, receptor undergoes conformational change. Steered docking, guided by additional constraints of the prior knowledge. Peptide docking, then protein docking. We have also reverse docking. Docking, how the docking? It can be between protein ligand, protein peptide, protein nucleotide. And these are the types of computer language that you will see when you will feed the field. So I don't know if you have done, then these things are understandable, but try to understand what is docking algorithm. These are key components of docking program: search and scoring, algorithm or function. The research algorithm positions molecules in various locations, orientations, and conformation with the active sites. So these are the few softwares. These are the algorithms, and we have scoring functions. So scoring functions will decide where the interaction with the ligand and the small molecule is what is the scoring function, and that will decide the functionality of the molecule, which can be taken further or not. So these are some computational terms. It is used. These are some softwares used for molecular docking. Like few are free. You can see AutoDock, vNice Dock, and FlipDock, and others are paid. a few remarks are there. Then you have web server where these softwares are there. How the docking is done. First you prepare a protein, then you prepare a ligand, grid is generated, their interaction is done, and we do post-docking analysis. This is just what I have told earlier. How protein is prepared. A typical PDB structure file consists of heavy atoms. The structure of protein is like that. And then these bond orders, topologies, and formal atomic charges are noted down. After that, you note down terminal amide groups, ligands, extra structures. These things are there. Ionization, tautomeric state. These are the values. And then something, when you open any software, something like this, that needs to be filled up. Then after that, you prepare first protein, all the values are filled up, then you prepare a ligand, to which ligand the protein has to be interacted. And this So when you prepare a ligand, the conditions are there. They must be three-dimensional. They must have realistic bond lengths, bond angles, torsional angle. They must have all the hydrogens, pinned valencies, and they must have an appropriate protonation state, physiological pH values. They must be supplied with how you feed in computer. Mastero, they are the files, Mastero, SDF, MOL2, and PDF format. So this is when you open the computer, something like this something like this will be there. Then grid generation, third step. Receptor grid generation requires a prepared structure. Something like this will come. Then finally, fourth is docking. The glide ligand docking jobs require a set of previously calculated receptor grid and one more ligand structure to whom need to be docked. Prerequisite for docking are prepared protein structure, prepared ligand, selection of right protocol. In case of a number of poses that needs to be generated. Like this, things will come. And finally, you have post-docking analysis. Here, typically protein ligand docking software consists of two main components. That is, one is search algorithm and scoring function. Search algorithm will generate a large number and as a characteristic of extensive sampling and different poses, limits duplicate visits. And when you score a function, that calculates a score or binding affinity for a particular pose, binding affinity for a score representing the strength of binding. So this is when you do in computer, something like this will come. Scores, these are the scores, these are the binding, predicted pose, like something based score. Then you have interaction diagram. I think how the things will look in computer when you perform it. Few applications, let us try to see. It helps in identification of hits. It helps in lead optimization. It helps in designing of more potent and selective analogs. And it helps in comparing different molecules, what are the affinity to the ligand of the molecule. It helps in understanding interaction, role of a specific amino acid in binding affinity. So, with this brief introduction about the pharmacoinformatics, let us try to see how does this pharmacoinformatics is going to help you. So you have traditional formulations filled with Ayurvedic knowledge. When you phytochemically profile them and perform in silico analysis and identify the bioactive compounds from traditional formulations, then you have a scientifically validated therapy. It reduces the time of research. Okay, it reduces the time and gives you validated therapies for whatever disease you are targeting. Here I have written antiviral and anti-infectives on which I am working here actually. So here also you can see the same thing in silico computational analysis. What it does, it narrows down the candidate of compounds by studying, with the help of molecular docking, with the help of quantitative structure-activity relationship, molecular modeling, dynamic simulations. You can predict and narrow down the candidate compounds. What do you predict? You predict the bioactive compounds and their interaction with biological targets. And that speeds up the research and development process. And these are some, you would always be requiring the help of this traditional wet lab thing, like analysis with chromatographic technique, phytochemical analysis, and this batch to batch control. All things are required. But in silico computational analysis is going to help you a lot when herbal analysis is concerned. A few gap areas that needs to be highlighted, like standardization of the formulations is major one, and the integration can solve the problems, followed by this. If we combine the if we combine the traditional knowledge with advanced technologies in silico modeling, nanotechnology, and clinical validation, it is possible to create innovative and effective plant-based therapy. So this is what is the gist of this lecture actually. So when you start working, then you will come to know. In brief, I have presented this topic. So over to questions, and if some questions are there, I would be happy to answer them. In brief, I have tried to do this. Yes.
Any question?
I think I have tried to cover how this pharmacoinformatics is going to help in development drug discovery from the Ayurvedic formulation part. The basic problem with Ayurveda is that when we talk of pharmacoinformatics combined with Ayurveda, it becomes Informatics. The information needs to be exchanged between the two, with pharmacoinformatics and Ayurveda. So until and unless the chemistry of Ayurvedic formulations are defined, there will be no exchange. And when you start defining the chemistry, you will find that a large number of molecules are there. So which molecule should we take forward? In that sense, pharmacoinformatics is going to help you that this molecule is better. Try to concentrate on this using traditional wisdom. And then you validate using that. It reduces the time. It will reduce your all animal study data, because these formulations we are using for ages. So in that purpose, when you combine both the things, though pharmacoinformatics is a tricky one, you have to learn a lot. You have to sit on computer always. You have to learn the computer language, how to feed the molecule, how to enter the data, how to analyze the results. But it helps actually. So that way. That's why this particular department is separate in NIPER. We are working in natural products with the help of pharmacoinformatics. We have created this field, bioinformatics, and we are trying to move further on. So this way, this presentation was all about. I think this will help you in understanding things. Over to any questions.
Thank you, Dr. Lal, for nice
Thank you,
presentation. And I think it is a very important, it will play an important role in development of different type of drugs. So,
Excuse me, sir. There are some questions are there.
Please.
So first one is, what exactly is bioinformatics and how does it differ from conventional pharmacoinformatics?
I think I have explained that in my opening slides only. Bioinformatics is a interdisciplinary terms. If you break bioinformatics into two, first one is Ayurveda and then comes informatics. So if you combine them, it is the exchange of information between two informative things. Like in informatics, you have the information about the molecule And in Ayurveda, you have the information about the traditional formulation, their analysis, their usage. So when you combine both, it becomes bioinformatics. And how bioinformatics is going to help in drug discovery, that I have discussed in this presentation. So it is different from pharmacoinformatics because there you deal mainly with the synthetic molecules. What you do in pharmacoinformatics, you have a target, you try to design molecules, then you select lead molecules, then you create a list of molecules that will be active. But in bioinformatics, it is different. You have a formulation, you have active formulations. You are trying to take a molecule from the formulations to forward, and that is going to help you. That is going to help you to further discovery of lead molecule from formulations. So it is already active, and in that reverse pharmacology and systems biology is also assisting because of it. Noted down this biological activity we are using since ages. So that is how bioinformatics is coming into it.
Thank you, sir. Now the another one is, can molecular docking and in silico modeling be effectively applied to phytochemicals used in Ayurveda?
Yes, exactly. It can be applied. Molecular docking in silico, yes, it can be. But what I showed you this slide only. It can be applied. What it will do is, I think it will narrow down the candidate compounds that I showed you in this particular slide. In computational analysis, along with phytopharmaceutical research based on Ayurvedic formulation, and with the help of the information obtained from molecular docking studies, quantitative structure activity relationship, and their dynamic simulation, we can narrow down the structures, number of compounds that has come active, and that we can always predict the bioactive compounds that interact with biological targets. And this will speed up the research. Speed up, and it will give you validated research, because already the formulations that we are using in traditional system of medicine, they are active, but since they are not standardized, for that purpose, people are not using traditional formulations, and this is going to validate the things. This is one of the important things, that is when we combine informatics with Ayurveda, this is one of the important this is one of the important help that we get from pharmacoinformatics in Ayurveda. Yes.
One more question is there, sir. In the next 5 to 10 years, do you force for bioinformatics in global drug discovery and development?
एक मिनट आप फिर से can you come again?
Sir, in the next 5 to 10 years, what role do you force for bioinformatics in global drug discovery and development?
In next 5 to 10 years, this is
Yes, sir.
with the help of pharmacoinformatics, definitely many more phytopharmaceuticals will be developed in India, actually, I can say, because you might have heard separate this phytopharmaceuticals, class of phytopharmaceuticals has been approved by CDSCO, where you have an active extract, only 4 molecules need to be quantified, and that becomes a drug. So in that case, the data generated by artificial intelligence or informatics will help in development of those drugs. And it is going to fill up the drug discovery process, speed up the drug discovery process.
Okay, sir. Thank you. Last one, sir. A
Yes.
protein drug can develop its efficiency and efficacy through bioinformatics technique. Is it possible?
Efficiency and efficacy can be predicted.
Yes,
That
sir.
needs to be tested in wet lab also. If certain molecules, suppose out of 100 molecules in the formulation, 5 to 6 molecules are very active. Okay, and that you try to enrich those molecules in particular formulations and keep that in market. So with the help of bioinformatics, you can have the enriched biologically active compounds. Okay.
Next one, sir. How do chem informatics and molecular docking help in validating Ayurvedic formulations?
Validating in terms of
Ayurvedic formulations.
Ayurvedic formulations. Okay. So now, how these Ayurvedic formulations are validated now? Abhi, in present scenario, how these Ayurvedic formulations are validated? They are validated with the help of the active components present in them after their analysis. Okay. So what this pharmacoinformatics will do? Those chemicals which have been analyzed as a major constituent in the formulations, are they biologically active for the same disease? So this information can be provided by the pharmacoinformatics, artificial intelligence, and that fills the gap between the modern system of medicine and the traditional system of medicine. And that is how the things, it can be validated. Pharmacoinformatics helps in quicker validation of the Ayurvedic formulations in terms of biological activity. So that way, it can validate.
Very good evening, sir.
Very
Sir,
good.
one more question.
Please.
Sir, can bioinformatics in terms of artificial intelligence, can it be beneficial in devising precision medicine in Ayurveda?
Precision medicine, you means for individualistic medicine?
Yes, sir. Yes, sir.
Pharmacovigilance type thing, na?
Yes, sir. Yes, sir.
So, those things, it can only help. It can only help.
Okay.
I mean, the precision medicine cannot be because Ayurvedic people, they have a different, they have different mode of diagnosis. They do some nadi prediction and all, so it can help only. It will, see, pharmacoinformatics or bioinformatics, it will only help in filling the gap between modern system of medicine and this traditional system of medicine. It in no way it will enhance traditional system of medicine or it will enhance modern system of medicine. It is basically helping to fill the gap between the two. Like Ayurvedic people, they do not understand what is biological activity. They will talk in their own terms. So the same term cannot be explained to the patient who does not know Ayurveda. So in that way, this informatics will help in understanding it. It is basically, it will help in filling the gaps between the two systems of medicine. That way, I don't know if I'm able to answer your query.
Thank you, sir. It was quite informative. Sir, one last question for you. Sir, Ayurveda is our holistic approach of Indian traditional medicine, and the Ayurveda is typically based upon a theory or a. So, can AI model these principles based on machine learning? Can these models be and any data paradigm can be modeled basing upon this Tridosha in Ayurveda? Can it be very beneficial?
These things are being tried, but I don't know how far these Ayurvedic people are getting success on this. Like, for example, certain plants, there is a Draviguna, something like this. For particular plant is having certain properties according to Ayurveda. Okay, like they are thick, they are something heavy, laghu, something like that. So some laghu plant cannot be used in if you have some intestinal disorder, those plants cannot be used. So what can be done? The information from Ayurveda can be combined for the treatment of intestinal disorder based on this type of thing. So like that, people are trying to trying to have information from, and they're trying to devise some artificial intelligence type of thing where the things can be taken from Ayurveda and applied to the informatics things. But how far they have succeeded, this is just a start. It all actually, informatics term itself was coined way back not less than 4 to 5 years. So that way people are trying. Okay.
Thank you, sir. Thank you for your question. Now it is time to express gratitude to all those who made this event possible and memorable. To deliver the vote of thanks, I am honored to invite a highly respected academician and guiding force behind this initiative, Dr. Siddharth Sankar Hotta, sir, Associate Professor, Faculty, CV Raman Global University, Odisha. Sir, we request you to kindly share your words of appreciation and formally extend our thanks to the dignitaries, speakers, and participants who have enriched today's proceeding. The floor is yours, sir.
Yeah, hello. Am I audible? Hello.
Audible. You can carry on.
Yes, good evening. On behalf of the Faculty of Pharmacy, CV Raman Global University, Odisha, it is my privilege to extend a heartfelt vote of thanks at the conclusion of this today's session. First of all, I would like to express my deep gratitude to Dr. Umaranjan Lal, Assistant Professor at NIPER, Sasnagar, Mohali, for delivering an enlightening lecture on this Informatics, that is the pharmacoinformatics in Ayurveda. Your insights into this blending of traditional Ayurvedic knowledge with modern informatics tools have truly opened a new perspective for our participants. I also take this opportunity to sincerely thank our Honorable Vice Chancellor, Professor Dr. Pansithar Mazishar, and our respected Pro-Vice Chancellor, Madam Professor Dr. Guda Sidevi, for their constant encouragement, vision, and inspiring presence that always motivate us to strive for excellence. My sincere appreciation also goes to our Dean Sir, Professor Dr. Amir Kumar Pushti, and all members of this organizing committee whose tireless efforts have made this event possible. A special thanks to our distinguished participants, faculty members, researchers, and students for joining us today and making the session interactive and meaningful. With this, we look forward to our continued participation in the upcoming session of the 6 days national webinar. Thank you all and have a wonderful evening. Thank you.
Okay, thank you
Thank
one
you,
and all.
sir. Now I would request dear sir to present a token of appreciation and honor to our renewed speaker. Sir, please do the honor.
Thank you very much, sir. Thank you very much. Thank you very much. Dr. Pushti is probably mute. Please tell him to.
Oh, sorry. I'm mute. So please accept the
Thank you very much.
online gift. So we will send it by post to you. So in the near future, we are also planning for collaboration and different type of groups in this field.
Please.
So I sure you will
I
help
hope
our
sir, my
students.
lecture would have been fruitful to all the students.
Right.
I don't know. Later on they can say. Okay.
Very nice. And Sangeeta, you can also share the certificate.
Sir, it's time to closing the final session. Respected speakers, honored guests, and dear participants, as we draw this enlightening session to a close, I want to take a moment to reflect on the journey we have shared today, a journey that has taken us from ancient Sanskrit verses to the modern day algorithms, from herbal wisdom to digital precision. Through the lens of informatics, we have witnessed how tradition and technology can harmonize, not in opposition but in collaboration. The presentations today have shown us that pharmacoinformatics is not just a tool for modern drug discovery, but a bridge that connects the timeless insight of Ayurveda with the evolving needs of global healthcare. To all our speakers, thank you for your depth, your clarity, and your to our audience, thank you for your curiosity, your questions, and your commitment to learning. And to the organizers and volunteers, thank you for making this session seamless and impactful. Let us leave here not just informed but inspired to continue this dialogue, to build new collaborations, and to ensure that the wisdom of Ayurveda finds its rightful place in the digital age. As the ancient text says, Yat Pinde Tat Brahmande, what exists within us mirror the universe outside. May our effort in Ayurveda continues to reflect that harmony. And namaste. All are requested to join us, same place and same home tomorrow. Thank you.
Thank you very much.
Thank you. Thank you.
Okay.
Recording stopped.